The broadcast is now starting. All attendees are in listen-only mode.
Hello, everyone. Welcome to the latest installment of the HDSA Research Webinar Series. I'm George Yorling I'm Director of Medical and Scientific Affairs here at HDSA, and today I'm really excited to have Dr. David Howland from the CHDI Foundation here to speak to us about the use of the presentation entitled Mind the Gap, Thinking Big to Bridge Drug Development in Huntington's Disease. And this will be talking about the use and development of different animal models to recapitulate HD. But before I turn it over to Dr. Howland, I just want to go over a few instructions just in case you're new to the research webinars. You can ask, and I encourage you all to ask as many questions as you like during the presentation. Just go into the question box on the right-hand side menu that it's highlighted here in red. So type it in at any time, and we will answer them upon the conclusion of David's talk. We're also recording this webinar, so approximately 1 week after today, you can all turn to hdsa.org /research webinar to see an archive of this, just in case you have to tune off early, or you know of others that would be interested in seeing this. Feel free to send them this link. And finally, just want to bring you up to date on our upcoming schedule for the research webinar. We will not have a formal webinar in June. Instead, we are excited to have Dr. Leslie Thompson from the University of California at Irvine, where she'll be joining us as the keynote speaker during the research forum in Louisville, Kentucky during our annual convention. We'll be live streaming that, so that'll be available for everyone to participate in. In July 17th, we're having a rescheduling of our April webinar where Dr. Ray Dorsey from Rochester is going to be talking about the Reach to HD PBT2 results coming out of Prana. And finally, in August, Dr. William Yang from UCLA will be talking about his latest data looking at different mouse models of HD and how he's using them to develop and identify and validate new therapeutic targets for HD. So with that, I want to just introduce Dr. David Howland. David got his PhD from Penn State and was an NIH postdoctoral fellow at University of Massachusetts, Worcester, where he investigated mechanisms of neuronal specific gene regulation. In 2006, he joined CHDI, where he is now the Director of Model Systems based in the Princeton, New Jersey office. At CHDI, David oversees the in vivo research portfolio, or the animal portfolio, which includes generating and characterizing new models for HD. He also oversees the target validation activities, which use these HD models, including this, testing genetically modified mice and pharmacological agents. Before joining CHDI, David worked at Wyeth Research, where he led target validation efforts in the neuroscience division. There he also developed and implemented a transgenic rat model for ALS, or Lou Gehrig's disease, and ran a program aimed at identifying therapeutic interventions for Lou Gehrig's disease. As a scientist at Cephalon before Wyeth, David co-developed rodent models of disease, including the very first knock-in mouse and first transgenic rat model for Alzheimer's disease. So with that, I'm going to turn it over to David. Presentation. Here you go, David.
Okay, just as acts with this.
Should be able to just,
Okay, thanks, George. Thanks for inviting me to give this webinar. As George said, I'm one of the science directors at CHDI, and what I'll be talking about today is some of the work that I'm doing with the in vivo model systems, including mice and higher species models, and their importance for implementation in drug discovery and research for Huntington's disease. So just for those real briefly that may not know what CHDI is, we are a private funded through Anonymous Donors Foundation. We are a not-for-profit with no competitors, only collaborators. That means we do have a wide freedom to operate with our mission solely to develop therapeutics for Huntington's disease. So that mission statement, as it's shown here, is to rapidly and collaboratively develop therapies that will slow the progression or delay the onset of HD. Importantly, we encourage scientific partnership. We like to think ourselves as collaborative enablers, where, as it says on the bottom here, really what we want to do is get as many people as involved in the mission to bring therapies for Huntington's disease. That includes university investigators, biotechnology firms, pharmaceutical firms, government, anyone, to get on this problem because we want to move quickly and as efficiently as possible. Part of our strategy also is to de-risk programs for pharmaceuticals. Pharmaceuticals have a very specific mission and a very specific bottom line. We want to encourage them to get into Huntington's disease research, get into clinical trials, and bring some of their valuable therapeutic small molecules, for instance, bring those to bear on Huntington's disease, and lowering the hurdles for them to get involved, to get interested in Huntington's disease. That's part of our mission and really an important component of what we try to do. So today I just want to kind of summarize very specifically where I feel and the foundation feels is the state of the art of using animal models in preclinical research to really not go very specifically necessarily into the individual animal models. I mean, I'm happy to discuss that at any time with anyone that wants to delve into that with me, but really to emphasize some of the pitfalls and hurdles that we're trying to overcome so that we can better use as a research community animal models in the early stages of an exploratory pipeline that helps bring leading therapeutic targets or ideas for clinical investigation. So this very simple scheme I'm showing here shows really there's two major components for any program that leads up to a clinical trial. There's the early biological exploration and validation, which is a long, laborious, tedious phase that scientists go through to get up to a point where something looks interesting enough and promising enough to begin to think about testing those in phase 1, 2, 3 clinical trials in the patient population. So as is highlighted on the left, that early biological exploration and validation includes scientists forming hypotheses, testing those hypotheses, and importantly, confirming those hypotheses, and that's something that we call validation. Once these hypotheses are firmed up and they're considered valid, efforts are made to actually develop drugs. That could be small molecule drugs or a gene therapy, for instance, a biologic or something that targets the Huntington gene itself, a Huntington-lowering therapy that most of you, I'm sure, have heard about, to test in the model systems for proof of concept that are these things going to work, and importantly, in addition to effectiveness in the animal models, to address safety. Are these things safe? What are the things we need to potentially look out for in the clinical setting? Once all these major categories are checked off, essentially, then one can start to think about entering into early phase 1 safety trials in normal individuals, and then eventually HD patients, and then phase 2/3 trials, which then actually addressing the effectiveness or so-called efficacy of a drug in smaller trials, phase 2, and then larger phase clinical trials to get those molecules out into the patient population after FDA approval. Now that's all well and good, but the failure rate, not only for Huntington's, but many neurodegenerative indications, is relatively high. Things do fail at a reasonably high rate in phase 1 safety, and potentially even a higher rate in phase 2, 3, so-called efficacy or effectiveness, meaning showing an effect in a patient. It is not a slam dunk. Things do fail at a pretty high rate, and I think most of you probably are aware of this from trials that have already been conducted in the HD clinical community. So there are many reasons for potential failures, but what I'm going to try to touch on today is, can we do a better job in this left side of my diagram in the biological exploration and validation? Can we do a better job implementing our animal model systems for validating approaches and strategies that are worthwhile, then will have a better shot at being efficacious or effective in the clinical population on the right side. That's what I'm going to try to touch on today. The good news for Huntington's researchers, at least, is that we know the causative gene that was cloned back in 1993 by a consortia of scientists. It's the so-called Huntington gene, a single gene mutation that has an expanded CAG track that 100% of HD patients have and ultimately will develop the disease if the CAG track is of a certain expanded length, essentially 40 or above. So that really is fortunate, at least in terms of what we can do with genetic models. Other diseases like ALS and Alzheimer's disease, only a fraction of the total population can be attributed to a specific gene or genes that people inherit. The great majority is called sporadic. So with Huntington's, 100% of the time we know the gene that's causing it. So we can use genetic engineering technologies to introduce these gene modifications, the mutant Huntington gene, into all kinds of model systems, into cells, into mice, into rats, even into larger animals. And that gives us a tremendous arsenal of reagents and tools, these animal models, to use to investigate exploratory biology, but more importantly, to identify potentially and promising therapeutic strategies. So this is just a schematic of an example of the Huntington gene. It certainly can be introduced into complex cell systems for scientists to study systems on a petri dish. Can and has been introduced many times into mouse models. But maybe some people don't know, there are existing sheep, Minipig, and non-human primate models of Huntington's disease that are now gaining more traction into the preclinical pipeline that I'll hope to touch on in a few minutes. So, but as I alluded to, there's a gap. And this schematic is meant to show the gap that exists on the left side between testing potential promising therapeutic agents in mouse models and then hoping that those will translate well to the human condition. That hasn't really worked out all that well. Mice are very good tools early on in our pipeline to gain traction proof of concept, but they may not be sufficient in isolation to have high predictability of what one might expect to see in the human condition. So there's certainly examples of that in Huntington and other diseases of neurodegeneration. So how can we as a foundation, how can we as a research community bridge this gap? So getting back to my schematic here of the biological exploration and validation on the left with all these tools that we have at our disposal, the cells, the mice, and potentially the sheep, the mini pigs, and non-human primate models on the left. I would think to say that we can use our mouse models in better ways, and I'll touch on that in the next few slides. The HD research community needs to come together in a more community-based way to implement these models with standardized approaches, and I'll touch on that in a minute. But importantly, emerging higher species models that are closer to human in size, brain structure, metabolism, are potentially very critical to bridge that gap. Also, as some of you may have heard or are aware of, CHGI is involved in an effort called Enroll HD, which I'm not going to go into details today, but this is a worldwide observational study for HD families. It has three major objectives listed here. One is to better understand HD to figure out the factors that are affecting disease progression that could give clues for new targets and new drugs. To improve the design of clinical trials and get them done faster, better, faster, smarter, as it says here, and importantly, to define best clinical care to improve the health of the HD participants and so-called best clinical practice. So really, efforts are happening on both sides of the pipeline, with Enroll HD, with human patients on the right side, and to better use our animal model systems for better prediction of what could go into patients, potentially in the context of Enroll HD. So in terms of how can we better use mouse models, well, we actually had a webinar several months ago with the JAX lab that some of you might have been on, but it was our describing some of this, so there won't be too much detail here, but it is our shared goal to improve the translation of results from the clinic and accelerate the pace of HD therapeutic research. But the challenge in the HD research community is how do we do that, and how do we use mouse models in testing specifically candidate therapeutics. As I alluded to, mice are small brain, and there have been numerous reports of positive results of certain drugs in mouse models that we need as a community to be very cautious with these data. It's exciting to see, but these are preliminary proof of concept results that need much more further study. They need A, replication from the first investigator that reported it. Can this now be seen by other investigators? Can this be seen in multiple mouse models? And more importantly, can this be seen in higher species models potentially? So we want to be very careful because the HD community patient population is a very precious resource, and jumping into clinical trials straight away from exciting but still preliminary mouse reports that appear in the literature is something we need to be extremely cautious with. So it's important to really validate these studies across labs and ensure that these results are legitimate, bona fide, reproducible, and robust, and then to extend the data that's published further into other model systems that are potentially closer to human to really understand if it's going to best predict what will happen in the human condition. So we set out, at least for the mouse work, to devise a field guide to working with mouse models of Huntington's disease, and this is really targeting research investigators in the Huntington community. This is a version of the front page of this manual, and the hyperlink that's shown below. It's available through the Jackson Labs website, through the CHGI website, and Psychogenics website. This was a manual that was written by these three organizations together, putting our heads together to essentially define a series of best practices. So it's an overview of the different types of mouse models. There's different flavors of mouse models that are out there. There's probably over 20 different types of Huntington mouse models that are available to the Huntington research community. They differ in ways that the mutant Huntington gene was introduced. There's fast versus slow disease models that exist. They exist on different strains. Black versus brown versus white mice, for instance, can affect the different phenotypes that are recapitulated in these models. So we summarize these differences, and we try to provide guidance on how investigators should choose a model for their specific studies. We also try to provide guidance on the optimal care and husbandry practice, how mice should be kept in their home cages, for instance. For instance, in an enriched environment is what we recommend, where mice are exposed to toys and tunnels and igloos so that they can hide, they can sleep, they can be cognitively stimulated. Not unlike a human condition where you don't have a mouse isolated in a cage, where people are out there, they have caregivers, that they're getting help. we want our mice to be experiencing that as well. We want our mice also to be co-housed with non-HD or non-affected mice, so mimicking potentially a human situation where it's a normal socialization and a normal interaction that occur. That this isn't necessarily done by all the research laboratories across the world, but we are promoting that these type of standard practices should be happening when testing candidate therapeutics. So these defined best practices are critical for labs to hone in on a defined set of mice using a common set of methods, which will greatly advance our ability to compare results from one lab to, for instance, in Iowa to results in another lab in Japan. We'll be able to potentially go towards that type of realization if people can begin to operate under these standardized practices. And I think this manual is a good this field guide is a good first step towards achieving that. These are critical things that the foundation believes are important to get the best possible data out of the mouse models for the best possible leads that can go forward further down our pipeline towards clinical advancement. So that's important. But is that enough? And this kind of alludes back to the title of my talk. It is going big, essentially. Where there's, mice are mice. They're small, right? So this is an example, for instance, of a human MRI, where certainly MRI can also be done in mice, as shown here. So adaptations to the MRI magnet and the software have been made where imaging can be done in anesthetized sleeping mice that are immobilized, where you can get a brain image. So testing potential candidate therapeutics in a mouse with an MRI endpoint is a valuable and a good thing to do early on proof of concept. But just how translatable, just how much confidence do we have that the result that we see in the mouse would be seen in a much larger human brain? Because as I said, humans are very different, much larger in size, whole body size, brain size, very different in metabolism, lifespan, brain neuroanatomy. It's a long way from a mouse to a human. So can we bridge that gap a little bit better? And we're in a good position in Huntington's disease that we potentially can. So here's just an example, right? So on the far left is the mouse brain. It weighs ½ a gram. Just to the right of that is a rat brain. It's about 2 grams. Just to the right of that is a monkey brain. And on top of that, you see an example of a sheep brain. And then to the far right, you see the human brain. So not only do you see a difference in the overall size, you could probably fit hundreds of those mouse brains inside that human brain, for instance. But there's a difference in the structure. If you look at all the convolutions and the folds of the so-called cortex, surface cortex in the human brain, that's a degree of complexity that's replicated at least to some degree in the monkey and the sheep. But if you look closely at those tiny brains to the left, those surfaces are very smooth. So there's a very different structure, neuroanatomy of the mouse brain and the rat brain compared to the sheep, monkey, or human brain. And these are critical differences that could impact how the disease presents itself in the brain of these different models, and we need to be aware of it. And I believe more effort needs to be spent modeling Huntington's disease in these larger animal models, potentially in sheep, minipigs, and non-human primates, to help bridge that gap in our preclinical research. So here's just an example. These things are happening. So these, on the left, there's images from an HD sheep, which have been generated down in Australia. This is just brain MRI that can be done and is being done in those sheep. And just example MRI from human on the right. And you don't have to be an expert in MRI to know that these images don't look all that different from each other. Other example, for instance, EEG. That's a non-invasive methodology to measure electrical activity in the brain. An example of this girl on the left that has a hat with surface electrodes, very non-invasive, very easy. Very difficult to do in mice. has to be very invasive by poking needles directly into the brain. So not very similar to what you can do in the human, but an example in a monkey that's shown on the right, which has surface electrodes applied to his scalp. There's a protective box over his head because monkeys misbehave and try to pull the electrodes off their head. But these are things that can be done. They can be done in sheep. They can be done in non-human primates. And one might expect that the results that you might see here are going to be much more familiar to what might happen in a human patient than doing similar things in that tiny little mouse brain that I showed you. So this is really just a schematic in trying to portray how we're thinking about it. So mouse models have a purpose. They're very, very important. They're shown on the left side. They're our first stop for our early therapeutic investigations. Early hints, as I say here, of an effectiveness of a drug or a gene therapy for Huntington disease. Early glimpse at, is it going to be safe or not? Should we scrap it? If it's going to be really toxic, if it's going to be a problem, we can scrap it right here. An early biomarker discovery, for instance. But that's not enough to leap over these two bridges I have to that human that you see on the far right. It's not enough, I don't think. We need to bridge. We need to advance the results that we see in that rodent model into potentially higher species models that are becoming available now to the research community. And there's all kinds of things that we can address here now with these larger models. Safety, biodistribution of a gene therapy. So if we administer a Huntington siRNA, for instance, into a mouse brain, well, the mouse brain is so tiny that that gene therapy will just flood the whole mouse brain easily. But remember the picture I showed you. Putting that in the human brain or even a primate or a pig brain, it's only going to cover a small part of it. And that biodistribution is very important to understand in the context of the large brain, the large human brain or animal brain, versus what you see in the mouse brain, which is tiny and it's not modeling well biodistribution. Mouse biodistribution will not model well the human condition. Brain imaging and EEG that I just showed you in the large brain that can be done in non-invasive ways in these large animal models, very powerful for preclinical research. And potentially even most important, looking at all the symptomatology for patients, chorea, deficits in their fine motor movements, cognition, apathy, depression, visual motor problems, the so-called saccades. Almost all of these are really very difficult, if not, let's be frank, potentially impossible really to well model in a rodent. You really need a higher brain neuroanatomy and structure to be able to recapitulate some of these symptomatic features, and even a subset of these are possibly best done in a non-human primate. For instance, fine motor movement of a species that has an opposable thumb and fingers, as opposed to a sheep that has hoofs and is four-legged. I mean, certain things you really have to potentially even go to a non-human primate to try to recapitulate. So what are the efforts that we're taking? So CHI is funding a global collaboration for both the sheep and mini-pigs, and we're considering efforts now to enter into the non-human primate space. So what we have going on for sheep is we have transgenic animals in Australia, as shown here. But we have a set of collaborators in the University of Massachusetts in Boston looking at Huntington-lowering strategies in those sheep. Investigators looking at the pathology in New Zealand, Auckland University. A group in Cambridge University, UK, behavioral phenotyping, doing EEG, for instance, and MRI in these sheep. And some molecular biochemical phenotyping also happening in MGH Boston. So you can see a worldwide concerted effort of people trying to advance this model for this pipeline that I'm showing you to advance therapeutics. Minipigs were doing something similar. It's not as far along as the sheep, but we have two collaborations, a group in the Czech Republic that's made an animal, and a group in Iowa that's in the process of making an animal. And phenotyping will go on and is going on at the George Huntington Institute in Germany, some molecular biochemistry in King's College with Jill Bates. Huntington measures in Biofocus in Leiden in Europe, and neuropathology, again, with the same group in MGH Boston. So good progress is being made. Some models are now just beginning to be used for testing in that scheme that I showed you for gene therapies that are actually starting to be put in these genetically modified large animals or programs that are potentially on a track to get to the human clinic. These are just some papers that I pulled up just to convince you that there's a lot of people out there that are publishing important data and information on the sheep model as shown in the upper right. The second one in the middle on the right side is a monkey model down in Atlanta, Georgia, and the Minipig model as well. So things are happening, and these models are emerging and beginning to be used in our discovery programs. So here's just an example of a Huntington lowering strategy. This is actually a normal non-human primate, but certainly these types of studies we hope will advance soon into genetically modified animals, where an siRNA targeting Huntington to lower Huntington was administered into the non-human primate brain, into a deep brain region called the striatum. And you can see it was administered on one side, and all the green and the bright yellow and the red colors administered where the drug went. And on the left side of the figure, you can see it wasn't put on that side of the brain, so you don't see any drug there. If you look at these little circles, this team actually went in after removing the brain and did something called punch biopsies, used a tool to excise little pieces of the areas where the drug went to ask how much Huntington is going down in the brain. And so they take these little punch biopsies and put them all into like little test tubes and do experimental assays and actually look at the degree to which the Huntington gene mRNA and protein is lowered. And you can see from different punches, you see different levels of Huntington reduction. So this is a good way of showing that you can lower Huntington in the brain of a large animal model. And it's important to highlight here that the degree of knockdown or reduction of the Huntington will differ depending on what part of the brain you're in. And the size of the brain matters. As you go further and further away from that circle, the level of Huntington knockdown will be less and less. These are experiments difficult to do in mice because it's pretty much going to be knocked down everywhere because, remember, it's so small that the drug goes everywhere. So the large brain is really critical for these types of studies to understand what's going to happen in the human brain. So these are real-time issues because there are Huntington lowering strategies being taken by several companies out there today. Most of you probably know about ISIS. There was a collaboration with ISIS and CHDI for years. That's now, we're very happy to report that that's transitioned now between a collaboration between Roche Pharmaceuticals and ISIS to advance Huntington lowering into patients. And they're doing toxicology studies in animals, not genetically modified animals. I think that's kind of past where they are right now because they expect to be in small trials in patients as early as 2015. Using therapies that have already been shown to be in early trials in ALS with so-called antisense oligonucleotides to be safe in phase 1. But there are good candidate programs needing these HG large animal models for testing that are coming along. A so-called Sangamo, which is out in California, Pharmaceuticals, has a therapy called AAV. This is adeno-associated virus. It's a viral therapy to direct a Huntington-lowering so-called zinc finger into the central nervous system of models and then patients. And there's a similar strategy that's being taken by Genzyme and separately a group at the University of Massachusetts and just outside of Boston that also uses this AAV viral-directed therapy, but their modality is called an siRNA. So suffice to say, zinc finger or siRNA, both are expected to target the Huntington and to lower Huntington. They just use different types of means to get there, but both require the delivery of a virus directly into the brain. So I'm happy to report that these types of therapies are now actively being tested in HD genetically modified animal models. So this is just an example of the HD sheep brain. As you can look where these yellow arrows are pointing, this is actually the AAV material that has actually been injected into the brain into a region called the caudate putamen. As you can see those white clouds, that's a contrast agent showing where the material went. You can see it's a very focused injection hitting one of the brain regions that it has been shown and suspected to be the root cause of a lot of the pathophysiology of Huntington's disease. So we want to have the gene therapy targeted here for proof of concept in the sheep to ask a couple of questions. One, is it safe? Do the sheep do well? How much Huntington lowering happens? And can we reverse some of the symptomatology of the sheep? Experiments that may be important to help guide best clinical trajectory of this type of strategy into the human patient. So this is happening now, and it's a real pivotal milestone for research and development, the use now of large animal models in addition to mouse models to investigate potential promising therapeutics for Huntington's disease. I think it's a real important transition the field is starting to take. So I hope I gave you a flavor of the efforts that are going on both sides of our efforts here. The clinical side with Enroll HD, and certainly we can provide more information on Enroll HD for those that don't know about it through the CHDI that can guide you appropriately to learn more about that. And on the early exploratory discovery biology side, and we're taking active efforts to how to better use mouse models early in the discovery process, and then how to transition at least certain therapeutic ideas into larger models to advance that proof of concept into something that's a little, that's closer to being what we might expect to see in the human condition, what we call translation, to improve that translation from the early pipeline to the later pipeline. So that's what the summary, let's just go through my summary. So improving the mouse models, and importantly developing the large HD models, which we think have high value for guiding therapeutic development, especially in transitioning into investigational studies. All these gene therapy studies that are ongoing now, that are coming to fruition, these large models are important for safety concerns, to address the safety of these entities before they go into the human condition, and the degree of Huntington lowering that happens in the individual brain regions that we think are susceptible and we're targeting, and potentially to see how much so-called effectiveness or efficacy we're seeing in these large animal models before transitioning into human studies. So we hope that these models can help in clinical trial design and predicting safety and success. And my little cartoon on the bottom really is trying to emphasize there needs to be, and there is, good communication that goes on between what's happening with our animal testing and how we relate what's happening in our animal models to what we see, most importantly, in the human condition. And what we learn in models can inform what we should be looking at in humans, but even more important, vice versa. What we learn from humans absolutely teaches us what we should be looking at and how we should be advancing these animal models. So with that, I think I'll wrap up and thank everybody, and I think George can open it up for questions.
Thanks, David. if anyone has any questions, feel free to type them in the box now and submit them. I'll go ahead and get us started. It's very exciting data to see that showed the monkey knockdown with, I believe it was the siRNA being administered to the monkey brains and also with administered directly to sheep brain. Maybe you can speak to the progress in the sheep. Are there plans to, obviously we can't do brain punches in humans to see if the lowering modalities worked. Are there plans in the sheep to test the newly developed CSF test to see if the Huntington lowering agents are lowering Huntington in the CSF or cerebral spinal fluid?
Right. So what George is alluding to there is a new assay called a Singulex assay that is able to actually detect the Huntington protein in a cerebral spinal fluid from patients. So these are exciting, very important data that have emerged, actually just a presentation at our recent CHI conference in Palm Springs. There was further validation of those data, so that's incredibly exciting. So obviously the priority has been to improve the sensitivity and validation of that assay for use in the clinical setting. That's the top priority. But what's also happening now is going back to our animal models and drawing CSF to see if we can detect the Huntington protein there. And that could potentially be used, as George, you're alluding to, as essentially a biomarker to investigate if we administer a Huntington lowering therapy like an siRNA. Can we look in the CSF of a large animal model, for instance, to see if that Huntington level is changing? So that would be a very important and valuable biomarker to say, yeah, Huntington is doing something. And I can report that at least in our transgenic rat model, using this assay, Huntington is detected in the CSF. So we have CSF collected from the monkeys, the sheep, and the mini-pigs. They're sitting in the freezer, and we're hopeful to get those assays run over the next 3 to 6 months.
Great. So here's, I know, David, you'll have a lot to say about this question. It comes up a lot in our HD animal model field. The question is the length of the CAG repeat track stable across generations in transgenic animal models?
Yeah, that's a great question. So the CAG track, the CAG, which is what we call pure CAG track as it exists in humans, is inherently unstable. That means it can either expand or contract, and expansion of lower repeats into higher repeats usually means more earlier onset for, well, essentially for Huntington's patients, it absolutely means earlier onset of the motor symptoms. So there's a direct relationship. The higher the CAG, the earlier the onset. No different in the animal model. So if the animal model was made with a CAG repeat that is essentially the same type of sequence that is in the human condition, the CAGs are unstable. They do show a propensity to change from generation to generation. There's a propensity to either contract to some degree or expand. The larger the parental size of the CAG, the more likely is that it will expand. This is an important feature of Huntington's disease, and it's an important feature of the models that our systems biology group now at CHI is capturing and really trying to understand the relationship of CAG size to the pathophysiology of the disease. So we're trying to use some systems-based approaches, some computational modeling approaches to understand really what is the overall impact of the expanding CAG, and can that give us insight into identifying new targets and therapeutic strategies. So yeah, the models are absolutely like the human condition. The CAG is unstable.
Another question, sticking with the CAG repeat motif, is what size are the CAG repeats, or the common size in a mouse model, and how long will the repeats be in a monkey if you're planning on creating a monkey?
Yeah. So a normal mouse running around your garage or your backyard will have 7 CAGs, or essentially 7 glutamines. Okay, and it's actually what we call an interrupted repeat. So it's not pure. There's a different code of sequence, a so-called CAA sequence that sits right in the middle. So CAA in molecular biology encodes glutamine, but so does CAG. So when you put that CAA codon right in the middle, it interrupts the track and makes it more stable. So the combination of a track of 7, which is very small, and a CAA in the middle makes it a very stable repeat in the mouse. So it's always 7q, no matter when you look, no matter how you look, it never changes. Now the transgenic animals engineer different types of CAG tracks of varying sizes. So the so-called R62 model has a track. Different flavors of R62 have different size CAGs. It's as low as 50, which is like an adult onset HD size. But there are models that have as high as 700 repeats. So investigators use these different size repeats for different reasons. So it's a complexity in the field that we need to be aware of. It's addressed in the manual, the HD field guide, pretty intensively. And it's a very important consideration to be aware of what is the CAG sequence and repeat size in your model, and how does that affect your disease phenotype. Now, monkeys have been generated outside of CHDI funding down at Emory University, just outside of Atlanta. This is a so-called rhesus monkey, where they use a transgenic approach. And they have about 80 some odd CAG repeats. So that's a juvenile size repeat. And they did it for a purposeful reason, because if you put a 40 or a 45 repeat in a monkey, well, you could argue that it could take 20, 30 years, 40 years to start to see robust symptoms of disease. And that, from a research perspective, might be unacceptable. So 80 is more juvenile size, and they expect a pathophysiology to develop over a 5 to 10-year time course. And they have publications that are showing that indeed is starting to happen. So that's the size they chose. Now, if CHI ventures into making further models, it's a point of discussion. I think optimally, you want to put different size repeats in different monkey models to be able to maybe have a high repeat to get a fast pathology, so you can use those in drug studies, but also have a lower repeat in the adult onset range, maybe 40 to 45, to really recapitulate the very subtle signs of disease, the very subtle changes that are subclinical that are happening to patients, like subtle MRI changes that you might want to recapitulate for investigational studies. So it's a good question and something we're wrestling with right now.
So another question, sticking with the large animals. If indeed large animals are so important for HD, why haven't they been developed before, besides the HD monkey at Emory?
The work has been going on for quite some time. So I think that there's two answers to that question. One is limitations in genetic engineering technology have made it much more difficult to make these types of gene modifications in large animals versus mice. So methods have been well worked out over decades of research to engineer mice. And even doing engineering in rats is not as easy as doing it in the mouse. Jumping into pigs, sheep, and primates is leaps and bounds much more technically difficult. So that's one reason. Second reason is, that from individual academic investigators, that the costs are very high. And the technology being difficult, the cost very high, I think has stopped some investigators from even thinking about it. But CHI, I think, values this very highly and has been promoting these efforts over the last really 5 to 10 years. And it takes a long time. Mice breed very quickly in large numbers, have short gestation times. It's 21 days from conception to birth of a new mouse, a set of mice from a pregnant mother, well, it's 6 to 8 months to get a monkey or a sheep to give birth. So these things add up, and it just takes a lot longer, orders of magnitude longer, to develop large models than mouse models. But it's been happening. And what I'm trying to emphasize today is the fruits of these labors are now being shown. These models are now, hitting their stride. They're there, and some of them are ready for certain types of investigational studies.
So there's a question about how everything you've presented about new, better, larger animal models for developing therapies to bridge the gap. There's a question about how what you've said might relate to an ongoing effort at UC Davis. I don't know if you're familiar with using stem cells to, and I believe this is what the question is asking about. The group in Davis is using mesenchymal stem cells as a delivery device for BDNF, brain-derived neurotrophic factor, to deliver to patients, and that would be a potential therapy. Would therapies like this be amenable to these larger animals?
Yeah, I think so. I think we're still pretty far behind, and it's just a matter of time and resources. But we're still pretty far behind on really describing all the let's call them phenotypic features that the models are showing us. We're still actively trying to understand the disease signs and symptoms in these larger models. It's been pretty well worked out over the course of decades for the mouse model. So for a specific transplantation or stem cell-based or BDNF-derived strategy that you allude to, there are certain things you probably could do in the large models, but it all depends on what you want to measure. In terms of placing the cells correctly, studying how well a cell graft survives, if the cells migrate further in the brain, is it safe, are there adverse events, certainly can probably certainly be done in the large animals, but if you wanted to measure certain outcome features that are specific to that program, those might have to be further developed before you can actually go do it. So I think there's some minimal experiments that you can certainly get your foot in the door, absolutely.
Great. When you presented the field guide for mice, I'm struck into how the similarities with HTSA has a physician's guide that they've created that we distribute to physicians for the best care of HD patients and distribute it around the world and country. What is CHDI doing to, the mouse guide is fabulous. I mean, what are they doing to distribute this and make sure this is in the hands of all HD researchers to try to get the best standardized care or treatment of mouse models? You can assure that CHDI-funded grants or contracts use these best models, but are people like the NIH adopting these standardized cares?
Yeah, so that's critical. So CHDI, by definition, for the researchers that we fund, we basically work with those investigators to follow these best practice guidelines. But, George, you're referring to the many investigators that are doing research around the world in mice that are not funded by CHI, and that's a lot of people. So we may not be able to convince everybody that this is the way to do it, and we don't necessarily have control, but it's a matter of getting the word out there and creating a groundswell in the community to say, this is the right way of doing it. And it's not only us that's saying this for HD, it's the ALS Association saying it for ALS, and multiple sclerosis saying it for that, and cystic fibrosis as well. So there are foundations that are really pushing on this. So for us, it's a matter of getting out there with these types of webinars, and getting out through our meetings and reaching out to investigators to do that. But referring back to the NINDS, there was an article written, I think probably 2 years ago, that we refer to in our field guide that actually speaks to this issue very much. Story Landis, who's the head head of NINDS, was a lead author on this article, essentially describing the problem of poor translation of the models into the clinic, not just for Huntington, but for many disease indications, and the need for standardized practices and standardized reporting back in the public literature of specific results. So the NINDS is taking this on as well and beginning to try to implement these practices into research grants that they see.
Okay. Awesome. Any questions here in New York before we sign off? No? All right, well, that's it. I just want to thank David again for his presentation, taking the time to come to our offices and present to everyone. Thank you again, and we look forward to hopefully joining us in Louisville or via the web for our research forum on June 21st.